So let me start by introducing Dr. Krzysztof Potempa. Can you hear me okay, Dr. Krzysztof?
Yes, I can hear you fine.
Great. So Dr. Potempa is a drug hunter dedicated to the prevention and treatment of neurological diseases that affects at least 1 in 6 people worldwide. He holds a Bachelor of Science degree in biochemistry from the University of Georgia and a PhD degree in neuroscience from the University Pierre Marie Curie in Paris. After working for a decade in pharma and academia, he established BrainCures, an award-winning data analytics platform company that leverages a novel biological intelligence approach to enable at least 60% drug development success rates without the need for deep learning associated with artificial intelligence. So, Dr. Potempa, please welcome and take it on.
Well, thank you for the introduction and good morning to everyone. I'll share my screen and present what the BrainCures platform can help you achieve to do. Well, thank you. So it's a pleasure to talk to you about what BrainCures does. We focus on novel approaches to precision medicine for brain diseases. So our aim is to deliver precision medicine for essentially 1 in 6 patients who are affected by a brain disease at some point of their life. And these diseases include ADHD, Alzheimer's disease, depression, epilepsy, pain, Parkinson's disease, or schizophrenia. And these are all diseases linked to synaptic plasticity, which is the really key focus of the key biology that BrainCures has mastered. And the reason why we've been focusing on synaptic plasticity since really 2003, I've been focusing on this, is the low success rates in the neurology field. Today, the industry average for clinical success from phase 1 to FDA approval is 1 in 10 success rates. In Alzheimer's disease, that success rate is just 1 in 25. Luckily, there is a growing market for brain disease, which will hopefully reinvigorate many pharma companies that have left the field to come back and do a warp speed operation Alzheimer's disease in the near future once COVID is tackled. BrainCures is we've been, you could say, operating in a stealth mode since 2017. We've received some recognitions such as being the best mental health Beanstalk Startup Award at the Giant Health Conference in 2017. We've really taken part in many accelerators and many either finalists or in the competition, and we've received some very good client and grant review comments. And just last year, Danny Sullivan from Longevity Tech featured an article which he called "Move Over AI, Here Comes Biological Intelligence." And you can look that article up, and it details how we've built a molecular GPS to nearly failure-free drug discovery. And my remainder of the presentation will focus on giving a few case examples and introducing really what biological intelligence is. So probably for most of the people here listening today, we know that biology is important in drug discovery. I think AstraZeneca has made it very clear with their 5R framework that if you don't have biology, you essentially fail. At least no clinical program without biology has really succeeded, at least for AstraZeneca. And when you have biology, AstraZeneca has been able to show that they were able to go from 4% preclinical success to 19% preclinical success. So biology is very important, and the biology that BrainCures uses is a molecular map that helps us eliminate preclinical failure enable at least 60 to 100% clinical trial success rates that are dependent on the protein type that the drug interacts with. And then we also shorten the drug development cycles by at least 4 years. And we do all this without deep learning steps associated with artificial intelligence because of the time and effort we took to figure out the biology behind learning and memory. And here I will play a 1-minute video of what biological intelligence is about. So, how is biological intelligence superior to artificial intelligence? Well, we know that with artificial intelligence, we usually start with a question around a compound, disease, or a data set. And we then have to go through deep learning steps that lead us essentially to generate questions about what our data haystacks or outputs mean. With biological intelligence, we have the BrainCures discovery engine, which I will talk about more in the next slide. And by using the framework that we've mapped, we're able to, you could say, identify the needles from the haystacks with the biology that powers the engine, the compounds that are associated with this molecular corporation that drives the BrainCures discovery engine. So I think it's very important to explain how we built our molecular GPS to preclinical and clinical success. So we actually started with, we first obviously chose the synaptic plasticity model, which has guided most of today's neuroscience drugs. And we've looked at the three main phases of the establishment of learning and memory, and we identified over 3,000 plasticity genes. We then performed the data mining step, which you could say the literature validation of the genes that we identified through a microarray analysis in the in vivo rat brain to identify 822 BrainCures discovery engine targets. We then used systems biology approaches such as network analysis and further literature mining and to define a proprietary algorithm that allowed us to separate the targets into what we call workers, managers, and directors of brain biology. And as we go up in this, you could say, molecular corporation hierarchy, we're able to obtain higher precision medicine potential. And I will give examples of how this works in the next slides. So I think a key question whenever one builds an algorithm is what is its predicted power to deliver significant translational outcomes. And there is a paper from GSK that identified, essentially mapped the drug target combinations that were linked to 72 phase 3 clinical failures. We use this data set to essentially perform with benchmarking of the BrainCures power to eliminate failure very early on in a clinical or preclinical program. And we were able to identify, when we use the BrainCures Discovery Hub filter, we eliminated 57 of the programs that then ended up going into phase 3 failure. This is, we're talking about at least $250,000 per each phase 3 failure. We're talking about quite a lot of money lost. So we're able to eliminate 57 phase 3 failures. And for these failures that we eliminated, you can see that for some, we at least eliminate 60% to 100 failure elimination rate, depending on the protein type, as shown here. So we achieve 100% failure elimination rates for growth factors and cytokines, and over 80% failure elimination rates for the G protein coupled receptors and transmembrane receptors, and also kinases. So I think our engine has quite powerful. Well, I think this data is quite indicative of the power of the engine to deliver high clinical success rates. So these would be at least 6 times higher than the typical 1 in 10 success rate. So I will focus on present a case study of some epilepsy work we've done with clients, and I think first to introduce epilepsy. It's a disease that affects more than 50 people a million worldwide. These patients often have a 3 to 6 times higher risk of death, and 1 in 3 epilepsy patients, the treatment is insufficient. I think epilepsy is quite unique because there's actually a lot of treatments, but still no cures. We have at least 3 ways of turning, you could say, data into knowledge. One is we start from genes, which we then pair with drugs using different platforms, such as even open targets. And we then do a prioritization using the BrainCures discovery engine, which we then take these outputs and can perform clinical novelty ranking to then define the next steps, or even identify what is interesting for your company in terms of the marketing strategy. So, obviously, some compounds, if there's the more knowledge there is, the less novel, I guess, that compound could be, but the less risky the development of that compound would be. I think something that's very unique to BrainCures is a virtual phenotypic screen, our pathway where we start with drugs and we go into the Broad Institute L1000 resource to identify the in vitro signatures. We then can apply so there are 98 genes that are common to the BrainCures discovery engine and the L1000, and this defines what we call the BDE virtual phenotypic screening opportunity, which allows us to perform essentially preclinical and clinical validation evidence, say phenotypic screen to identify interesting compounds for your diseases, especially if they have a component that links to synaptic plasticity. And a component that is, say, a bit more futuristic is today we understand that some of the compounds that modulate, particularly our masters and some of our hubs, because essentially act as bioanalytes for determining essentially if that compound maybe could be replaced, and then suggest specific treatment regimens. And so we are today able to typically if at a hospital say, please analyze these specific analytes, and then when we have the outputs, we can say, well, these would be the treatments that the patient should take and to achieve health again. So in one of the projects, we had the client provide us 10 epilepsy genes. We then performed essentially a 4-tier analysis where the genes were fed through the BrainCures discovery engine, and then we performed clinical novelty assessment, network connectivity assessment, and biomarker literature assessment. This allowed us to find that only one of those 10 genes was a BDE target. So this is what we call a primary BDE target. Then there was 5 of the remaining 9 epilepsy genes that we had what we call in the sequence a feature that qualified it as a BDE secondary target. So all in total, we then actually use these 4 features to define a BDE target score, and the top target that came out is actually the clinically most relevant epilepsy gene. So it's you could say in a way a validation of the engine's power to give relevant meaning, and we also from this analysis found that 5 of the genes that are listed here require clinical validation. They have already some preclinical data, and so if someone was interested in novel drug development, they'd probably focus on those 5 genes. The 5 remaining genes actually had both preclinical and clinical data, so they were maybe probably less novel for someone, good for someone who could be interested in repurposing with some evidence. But and then we also identified over 381 epilepsy biomarker publications linked to these 10 genes for the client. So this is how we could go from epilepsy genes to biology, intelligence-driven precision medicine. I think from the feature I'm most excited with, and actually many of our clients tell us that they like this feature the most, is what we can do if you say to us, well, we're interested in this disease, can we go from drugs to recommendations? So one way to do that is to go take the disease that you're interested in, look it up in open targets. And for epilepsy, we found that there are 67 investigational drugs. And what we did as the next step was we went into the L1000 resource and found that 34 of those 67 epilepsy drugs contain, essentially we identified 1,016 L1000 molecular signatures associated with those 34 epilepsy drugs. And the L1000 resource essentially is a big phenotypic in vitro resource based from the Broad Institute that's been used quite widely for repurposing and other drug discovery, phenotypic drug discovery efforts. That's the connectivity map, you could say, the next generation of the connectivity map. And so actually the BrainCures discovery engine, it contains in vivo data. So we're able to essentially cut down, superimpose the BrainCures discovery engine signatures to identify the molecular profiles of the compounds that have the L1000 signature. So it's a double virtual phenotypic screen. And we were able to identify essentially a number of drugs that had a higher BDE 100 molecular signature score than the current treatments for epilepsy, which are levetiracetam and carbamazepine. And here in this table, we actually list the 17 drugs we identified. And say which type of modulation it has, the compounds. So there's some compounds that have mixed modulation phenotype, and some have more of an excitatory fixing, and some have more inhibitory action. And for some of these compounds, there is the data saying how they contribute to the survival time. So, for example, that data is only available for 3 of these 17 drugs, but carbamazepine, for example, gives the longest survival time for an epilepsy patient. And so there are at least 4 different drugs here in this area that could be of potential interest for exploring at least how they perform compared relative to today's, you could say, state of the art. And I think a key point is that these BDE 100 prioritized drugs and their pharmacophores open avenues to biology and data-driven precision treatments. For example, drug X here, when we go into the literature, so we did a literature validation of the drugs classified by the virtual phenotypic screen, and we found that it has been shown to have very good safety and efficacy in 8 diazepam-resistant epilepsy patients, and doesn't show any adverse events except for a transient decrease in systolic blood pressure. So this is a drug we're talking with some KOLs to explore a slightly bigger, you could say, phase 2A study. And I think another feature I think that we're interested in when doing drug discovery is prioritizing patients based on biomarkers. So the Genomics England panel app allows a very good start for many diseases. So it has a panel app for epilepsy that has over 568 epilepsy biomarker genes. Sometimes because of cost interest, we want to actually reduce the number of those biomarkers, or at least revalidate them in a different way. And the BrainCures discovery engine allows us to do that. We're able to essentially prioritize 61 of these 568 Genomics England panel app genes as biomarkers, and we are able to essentially eliminate 39 of. So there's a traffic light system, which where the top red means that there's not so much validation information. Orange, there's intermediate validation information. Green, that means that it's a ready biomarker. So with this approach, you could say, added further, altogether 13 further epilepsy biomarkers that could maybe shift into a high clinical readiness with appropriate experiments. And some examples of the biomarkers identified include the BRAF V600E mutation, which correlates with multiple seizure types in the Chinese population, and also the KCNJ11 C166F mutation, which is associated with severe generalized epilepsy syndrome. So I think this is more of an integrative slide of what the BrainCures discovery engine can does all in total. And here we actually started with a drug pipeline where we looked at 207 drugs, and these drugs were then paired with 208 genes. And our BrainCures, you could say, prioritization actually identified only 20 of those drugs are linked to 51 target pairs, have you could say good potential to become precision medicine potential for for synaptic plasticity linked disorders. And then to add a good, you could say, validation context for epilepsy, we looked at the epilepsy literature and we identified the novelty risk assessment of the compounds. And we do that by calculating a clinical novelty index, which the higher it is, the more novel a compound is. So the current treatments have a clinical novelty index between 8 and 13, and some of the targets we identified have are a lot more novel and present interesting opportunities for further clinical development. And here I show actually the panel app of how we are able to identify, essentially select patients more precisely. And in the end, we found 31 target clinical biomarker pairs, and the client is doing some prospective experimental R&D on some of the predictions we made for them. And so I think a key point to say is that a lot of the biology is conserved, and our approach can also be used for things such as, for example, coronavirus target prediction. Here we shall just summarize some of the work that we've done in this space. We've gone into, as I previously said, Open Target is quite a good platform to start with. We identified 266 coronavirus-linked genes so this is first the data collection step. We then used our BrainCures discovery engine to do a knowledge extraction step, and this allows us to identify 172 drugs that are linked to 31 BDE targets. So 31 of the 266 genes are within the BrainCures discovery engine. We then did a filter relative to phase 4 ready drugs, and we've identified 107 phase 4 ready drugs, and they're all linked to 14 BDE target genes. And here I show what the genes are and some of the scoring. So we obviously are, if anyone is interested in this, we'd be happy to discuss the identities of these compounds and targets. So I think to, you could say, summarize the presentation, we've developed a very powerful approach to essentially turning your questions around compounds, diseases, and other data into better, safer, and faster drug discovery all across the drug development cycle. We have at least 3 different approaches, which is obviously using our BrainCures discovery engine. Then we also have the virtual phenotypic screening, the BDE 100-based virtual screening approach. We've also, I think, discovered through some collaborations with pharmaceutical partners that bespoke discovery engine building is something that's quite powerful, meaning that once you define the disease, we build another layer of that. We find complementary data sets, or the company sometimes generates a complementary data set to what we have to allow us to do the virtual screening and also apply our proprietary target hub master, you could say, algorithm to nominate those targets for the client. And today we're essentially looking for partnerships or investments to advance our internal R&D or strategic R&D strategies. And so this is both, we've got the data analytics platform that's ready, and in the future we look forward to developing our own pipeline and advancing precision medicine for brain disease and And yes, so I'm very privileged to have a great team that's supporting BrainCures efforts, and that includes Jean-Francois Williamson, who has over 15 years of experience as a biotech facilitator in this field, and then Peter Ball, who has 30 years of experience in bringing new products to the market. And one of our advisors is Professor Jacques Mallet, who's a distinguished scientist in neuroscience and gene therapy since the 1980s. And so be happy to take any questions. Thank you.
Wonderful, wonderful. Thank you so much, Krzysztof, for your insightful presentation. There's a question from Dr. Steve Miller from Sandia Biotech. He's asking, are you doing electrical activity studies using censorship technologies that map in real-time neural electrical activity?
Well, so the platform is based on long-term potentiation, which is a electrophysiological paradigm. So we have collected signatures, and the literature we most of the time look at is linked to that. So you could say, I would say yes, or I'd be happy to discuss further to understand better the specificity of the question.
Yeah, sure, absolutely. We can connect you with Dr. Steve after the summit. And Dr. Debano, do you have any questions or remarks?
Yes, I actually do. That was a very nice talk, Krzysztof. So I did have one comment and one question. So the comment is that the VRAF kinase V600E link that you found is really interesting, and an anecdote on that is that the drug against it was discovered by Plexicon, whose founder was my postdoc advisor when I was a postdoc at Berkeley. It's Plexicon and Sang-Ho Kim. So it was very nice to see it coming up again and again in different forms. That was one general comment. And the question was that I was going to ask you about applying your methods to other target families, other biology. And I see that you addressed that in your bespoke discovery engine. And I'd like to touch base with you on that regarding my other company, Exposed Therapeutics, and
Yes.
see if there are any collaborations we can explore.
Well, thank you very much. We're very open to and very flexible in how the collaborations can work. So
Great.
Wonderful. Thank you again, Krzysztof, for your
check with Sam on or Sherry in getting your email address.
Absolutely, yeah. We'll definitely connect you with Krzysztof after the event. And again, do want to thank you, Krzysztof, for your time and sharing the insightful presentation. And now, ladies and gentlemen, we will be going to our next presentation, which is discussion and demo of the CDT drug discovery platform. So if we can all head over to the next room, I would appreciate it. Thank you.